Okay, welcome to this ACAR at ANU student presentation session, everyone. I'd firstly like to acknowledge and celebrate the first Australians on whose traditional lands we meet and pay my respect to both the elders past and present. So my name is Max Kirkby and I will be chairing today's session. Our first presenter this afternoon is Veronica Padilla. Veronica, please start whenever you are ready.
Sure, thank you, Max. I'll just share my screen. Alrighty. Well, good afternoon, everyone, and thank you to the Australasian Council for Undergraduate Research for providing this wonderful opportunity. To introduce myself briefly, my name is Veronica and I'm completing my honours at the University of New South Wales in Sydney as the final leg of my biotechnology degree. And I'm here today to present a research project that I undertook at the end of last year. But before we get started, I'd like to acknowledge that I'm presenting today on the land of Gadigal and Bidjigal peoples, who are the traditional custodians of the Sydney coast. I would like to also pay my respects to the elders past and present and extend that respect to other Aboriginal and Torres Strait Islanders who are present at the conference today. So, what makes you unique? If we look deeper on a molecular level, nearly every cell of your being contains a sequence of DNA that is unique to you. DNA is your genetic information and is responsible for many of your physical traits, including eye color, skin tone, and in some of us, an unfortunate intolerance of dairy products. Importantly, DNA can also impact one's predisposition towards developing certain diseases. When most people think of DNA, something like this is typically visualized. This is called a double-stranded helix and has a basic structure that resembles a ladder that's been twisted over and over again. So much of what has been learned thus far about cell function and genetic inheritance has relied on the assumption that this structure is present. However, there are a handful of other shapes and conformations that DNA can adopt. And it's essential that we account for these alternative DNA structures whenever we try to make links between someone's genetic makeup and their healthcare. The target of my project was the intercalated motif, abbreviated to I motif. Looking at these schematics on the right, we can see that the I motif is quite distinct from the double-stranded helix. For example, we see more of a looping pattern as opposed to the twisting. If the entirety of human DNA were mapped out, I motifs would be enriched in certain areas. These areas are closely associated with aging, as well as cancers and neurodegenerative diseases, both of which are growing problems in countries like Australia, where we have a rapidly aging population. However, the study of I motifs is relatively juvenile, since it was only 3 years ago, in 2018, that they were directly observed inside live human cells for the first time. Last year, an estimated 50,000 Australians lost their battle with cancer, while the group of neurodegenerative diseases referred to as dementia was reported by the Australian Bureau of Statistics as the second leading cause of Australian deaths. By this measure alone, the gaps of knowledge in what we currently know about I motifs should be addressed, since they hold the potential to save lives. However, in the past few years, since the 2018 discovery, there remains only few tools in use for studying I motifs inside cells. Therein lies the need for more research and the purpose of my project. So the tool used in the groundbreaking 2018 research study was an antibody fragment. Now, an antibody fragment is a modified antibody that consists of just the parts which are needed to suit its purpose. In some cases, a smaller fragment is more advantageous than larger ones. A nano-sized format generally confers better yield, solubility, and utility, which is precisely why I set out to take the probe from that 2018 study and reformat it into a nanoscale framework. So my project's aim was twofold. Firstly, to produce a nanoscale antibody fragment that could target I motifs based off the larger 2018 version. And secondly, to characterize this new probe's specificity against a broad range of DNA structures in order to prove that my probe could still detect I motifs. The following materials were prepared or purchased prior to the commencement of my project. Firstly, bacterial DNA in the form of a plasmid, which is commonly used in science to produce proteins. And the specific plasmid that I used contained the nanoscale format for my probe. Secondly, the gene insert, which is named IM2B7HF, is the unique sequence that provided the 2018 probe with its specificity for eye motifs. And that's what I was trying to produce at nanoscale. And finally, E. coli, which are common and relatively safe bacteria to work with, provided the necessary biological machinery which was required for the synthesis of my nanofragments. I spent roughly 7 weeks working through the different stages of this project. Firstly, the preparation steps saw the recombination, which means joining together of plasmids, and the gene insert. I then transformed E. coli with these recombinant plasmids, which means that the plasmids were now sitting inside the bacteria. Then I had to add specific ingredients to the media that my E. coli were growing in so that they had the optimal conditions to produce my nanoscale fragment. And following that, I isolated and purified my nanoscale fragment and removed all of the other contaminating E. coli materials, leaving behind just my probe, which I finally applied in several tests against different DNA structures to see if the probe was able to target eye motifs. So to address the first part of my aim, let's look here on the left at my little figure of a gel. This contains 3 samples and a size marker just to the right. So if we're looking at the leftmost column, which I've marked in red, this column contains our nanoscale antibody fragment at the rectangle here at 17 kDa, kDa just being a measurement of protein weight. We can see that there is a second band just below, and that's actually another protein that was present in our final sample. I believe that this protein's presence was due to size limitations of the purification technique that I used. However, after consulting with my supervisors, we were satisfied that the main protein in our sample was the antibody fragment that we wanted, and that we had achieved a relatively pure sample of our fragment. Just looking to the right, these 2 lanes here are negative controls, and they provided the results that we were expecting. In lane 1, we have flow through from a filtration technique, and as you can see, it's empty and it's supposed to be void of any bands, which indicated to us that during filtration, we conserved all of the proteins and only allowed the liquid components to flow through, which is what we wanted at that stage. In lane 2, we can all hopefully see the numerous bands, which represent the unwanted proteins that we had successfully removed from our final sample. So we applied our sample of a nanoscale eye motif probe to a series of antibody-based assays, where the main principle at hand was to mix the probe with different DNA structures and see if the probe would stick or bind. If the probe did indeed bind, then it meant that the probe had some specificity for that particular DNA structure. If lots of binding happened, that would indicate that there was a strong specificity for that DNA structure. So looking at this graph over here, I've grouped in green different eye motif structures. They all come from different sequences that are found in the human genome, while in red, we have non-eye motif structures. So if we compare the green to the red, binding was most frequent whenever we mixed the probe with eye motif structures, while there was minimal binding occurring between our probe and non-eye motif structures, as indicated by the much smaller size of the red columns. We consider these observations collectively as strong evidence that supports our conclusion that our nanoscale probe has the ability to specifically target eye motifs and exclude other DNA conformations. We therefore concluded in this study that the results obtained determined the capacity of a nanoscale antibody fragment to be successfully expressed and characterized to target DNA eye motifs. And through its application in cancer studies or research of neurodegenerative disorders, this probe may elucidate the role of the eye motif within the cellular environment. In this way, a novel research tool such as the probe I produced may reveal new strategies that improve patient outcomes or at least enable further exploration of the genetic code. And this project wouldn't have been possible without my supervisors, Marty and Marcel. So I just wanted to acknowledge their contributions, as well as the University of New South Wales. And with that, I will end my presentation. And thank you all for listening. And I'll open the floor up for any questions.
Thanks very much, Veronica. There are now 5 minutes for Q&A. We'll begin with our judges. Judges, do you have any questions for Veronica?
Thank you so much, Veronica. I think you did a great job of condensing down such a technical and process-oriented experiment that you managed to do through your research. I particularly thought it was really well communicated how you went through at the start of your process to identify in literature different techniques and different biological materials that would sort of set you up for success with this. I know you said that this is a reasonably new and novel approach. Were there any other before you decided on that approach, was there any other alternatives that you explored or maybe could have utilized? And did you have any testing or reasoning as to how you ended up with the process you decided?
Yeah, so there have been several techniques that have been used decades in the past to study eye motifs, but it's always been it's always been outside of the cell context. So those techniques were like NMR, CD spectroscopy. So a lot of them are kind of like just shoot light at it and see what results we get. However, obviously, if we're trying to study something within the context of the cell, just shooting light at something, it will be interfered with by other lipids or the cytoplasm, everything that has to do with the cell. So it's not quite applicable, but I did go through those techniques just to see what has been done, and I guess just to brush up on what discoveries were made using those techniques. What specifically led me to adapt a previous probe into this nano-sized fragment. A lot of it had to do with my supervisor. He was the main researcher who developed the 2018 probe. And so when he and I were discussing, I guess we were looking a lot more long-term, and we were looking about how what could this mean, and look at all of the potential for this, but we're missing the tools. How can we do this? And we were talking about just in general the versatility of antibodies and antibody fragments as a research tool. And so that's kind of just how I led up into using a nano. I think the technical term for it is a nanobody, but just, to make it a bit more understandable would be like a nanoscale antibody fragment.
Thank you so much. And thank you for sharing a little bit of that collaborative insight and having that discussion as well. That's really interesting to know. Thank you.
Okay, thank you.
That brings to a close our 5 minutes of Q&A time. Thank you very much, Veronica, for the presentation. We'll now move on to our next presenter, Gemma Jeffrey. Gemma, feel free to begin whenever you're ready.
So my talk's on something quite a bit different to Veronica's. I think we might have been put together as the leftover pieces of science that nobody could fit anywhere else. And just a little bit of housekeeping first. If you have a picture of me currently floating around your screen, you can leave it in the top right-hand corner of my slides, and there will be nothing important behind it that it's obscuring. My work is on a phenomenon called El Niño Southern Oscillation, which I'm going to shorten to ENSO because it's quite a bit of a mouthful. And that's a thing that happens in the tropical Pacific that swings between extremes every few years and impacts weather around the world. And I've been trying to understand the dynamics behind these swings. So I'll start somewhere we're all quite familiar, which is weather and rain. And this affects all of us from day to day. We don't need to know, do I need to bring an umbrella? Is it going to rain on me? Is it going to be too hot today, etc. And we can find forecasts of the next week or so on the Bureau of Meteorology. But they only last a week. And we all know that if you ask the Bureau of Meteorology, is it going to be raining next weekend? They might be wrong, and they have been wrong, and your picnic gets cancelled. So it's really difficult to actually get these forecasts right. And it gets even more difficult as you go further out in time. So you can manage a forecast for the next week, but a forecast for the next fortnight is difficult, and trying to predict what the weather will be like next month is really hard. Trying to predict what the weather will be like on any one day next year is basically impossible. But what if we could predict the rainfall next year? So I've just said that we can't predict on any one day what will happen, but you can predict if it's going to be a drier year or a wetter year than usual. And even though that's a fairly vague prediction, it can be helpful. So Australia is the land of drought and flooding rains. And if you can predict if it's going to be a somewhat wet year or a somewhat dry year, you can predict if you're more likely to have drought or more likely to have floods. And working out the chance of these things can help you mitigate the damage that they cause. Similarly, if you want to know how much wheat or how much of a crop a farmer should plant, it's useful to know how much it's likely to rain over the next year, so what the water resources will be available to water that crop. ENSO can split years into El Nino years and La Nina years. So I've got here a plot of the rainfall during winter and spring in several La Nina years on the left and several El Nino years on the right. And you can see that the La Nina years have above average rainfall across much of Australia, and the El Nino years have below average rainfall down all of Eastern Australia. But what does El Nino and La Nina actually mean? So you might have heard that last summer was a La Nina event, and that means that the sea surface temperature in the equatorial Pacific looked like this, or at least the anomalies did. So this is the difference between how that point at the sea surface normally looks and what temperature was last summer. And you can see there's a patch of cold water in the equatorial, central and eastern Pacific. And then there's patches of warmer than usual water to the north and south of that, and over here near Indonesia and Australia. This means that in general, there is a more net flow of warm, wet water towards Indonesia and Australia. And so you get more rain. During an El Nino, you have warmer than usual water in the central and eastern Pacific and cooler water over near Australia and Indonesia and to the north and the south. And the result of that is you get less rainfall over Indonesia and Australia. So you have these drier years. This is one particular year and it changes from year to year. So this particular La Nina pattern is not the same pattern you might see in a different La Nina. And it might occur at a different time or have different rainfall impacts across the world. But there are generalizations you can make between these events that help you understand them. The simplest of these generalizations involves drawing a line across the equatorial Pacific and then taking a slice down into that water. And in that water, you have a warm surface layer and then a fairly sharp transition into a cooler, deeper layer. And this transition is called the thermocline. So if you start with a La Nina, you might have cooler water in the Eastern Pacific because the thermocline is shallower. And so that cool water is a little bit closer to the surface, it can mix better. And those stronger winds and more rainfall associated with the La Nina mean that you get more water building up. After a while, this can slosh back towards the Eastern Pacific and get rid of that cold anomaly. And you end up with a warm anomaly or an El Nino event. But then, because the warm water is spread out across the Pacific, it can radiate heat to the atmosphere faster and you get less warm water in the Pacific, which makes it easier for a little bit of cold water to slosh up towards the surface and get a La Nina event again. This model uses two main parameters. So surface temperature in the Eastern and Central Pacific region, which I averaged across this grey box here, which is called the Nino 3-4 region, but it just says, are we in an El Nino state or a La Nina state or somewhere in between? The other data that this model uses is the thermocline depth, so how much warm water there is averaged across the entire Pacific. And that's sort of a measure of the potential to begin an El Nino or a La Nina event. we used a data set with 96 ensemble members, so 96 different approximations of reality, because there aren't enough observations available of what the world looked like to know exactly how it was. So we have these ensemble members that all try and work out what the world was. And so we have 96 realities to fit the model to that give an estimation of the error. For each of those ensemble members or realities, we wanted to find what model parameters fit the data best. So for any given set of data, I've just picked some dots here. You can try and predict forwards using this model to each of the next dots. And you won't be exactly right because it's a simplified model of a very complicated system. But you can try and find what the numbers are that minimize the square of these distances or the square of each of the difference in the predictions forwards. And that gives you the parameters that best fit that model, or that best fit that data and say what the best model is for that particular set of data. Using the recharge oscillator model gives you two main parameters coming out of it. So you have the period or time between each El Nino and then to the next El Nino, or La Nina to the next La Nina, and a half-life, or a measure of how long the model can hang around and be valid for instead of decaying away. So the fits across each of these ensemble members or realizations give you results looking like this. The first thing we notice here is that results are clumped together. So even though it's not a precise normal distribution, I've been calling it normalish, and it's not an unreasonable approximation to say that the data is distributed normally and that a least squares fit is therefore a reasonably valid way of approaching the problem. The other thing you can note is that this circle here, there, and that one there refers to what happens if you average all of the realities and then fit a model instead of fitting a model and then averaging the realities. And you can see that trying to calculate period by doing that gives you a reasonably valid outcome. But if you were to calculate the half-life or sort of the persistence of the model throughout time for the ensemble mean, or averaging everything first instead of calculating it and then averaging it, you get a very different answer. So it's important to look at each of the realities rather than averaging the realities and then trying to fit a model. The other thing I can note is that the triangle shows some previous work that other people have done, and they have similar numbers to what we found, but not exactly the same. This is reasonable because, as I've mentioned, every different El Nino or La Nina event represents itself differently. And so over time, you have these changes in the underlying dynamics. Not necessarily, but over time, you have these changes in how it shows up and is represented. So if the Burgess et al. Team were using a different data set or data from earlier, they would get different numbers. So I wanted to look at how these numbers changed over time as well. So the black line shows the median of a sliding 10-year window fit to the parameters, and the blue box shows the middle half of the period or half-life over that 10-year window. And that's a measure of how variable these numbers are over time. And what we can see if we do this is the period changes really suddenly around 2000. And it changes outside the range of the blue box variability. So something happened around 2000 that changed how ENSO works. People have seen this before and how often events crop up, but nobody's really sure why. So trying to understand the physics behind it that gives us this period changing in the half-life could help us understand what happened that meant ENSO changed. And that could potentially help inform more complicated models that can do better predictions. So our data set gives similar results to what previous studies have shown. And it also shows that the underlying dynamics behind ENSO changed around 2000, which is not a result you can see if you just count El Nino or La Nina events, because there aren't enough of them really in any given decade. And because that could just be stochastic or random variation in the events.
Thanks very much, Gemma. Your time has come to a halt. That was an excellent presentation, and there are now 5 minutes for Q&A, and we'll begin with our judges. So judges, do you have any questions for Gemma?
Thank you so much, Gemma. I really want to commend you on your ability to sort of upskill the audience on some of those processes related to climate and El Nino and La Nina, so that we were able to sort of understand your like sampling needs and some of that rationale behind it. And I just wanted to ask as well, I know you were saying that this research contributed like a little bit to like a bigger, more complex sort of process that could come from this. And when you had that region selection, I think you said it was like the 3 and 4 like quadrants. Sorry, I can't think of the term you used, but for that temperature that you focused on, was there a like reason that you selected that based on the like parameters you were testing, or is that generally what people in the
Funny
field?
you should ask. So there are 3 main regions used. There's a Nino 3 region and a Nino 3-4 region and a Nino 4 region, which just move across the equatorial Pacific. So Nino 4 is basically Central Pacific, and Nino 3 is jammed up next to South America. And literature generally says that if you use Nino 3-4, you get the best representation of when an event occurs. But I did actually test all 3 of those regions, and they do give quite different numbers for the period, which comes down to that fundamental difference between different El Nino events happening kind of clustered towards the middle or clustered towards South America, and they're all different. So you do get different results depending on where you choose.
Yeah, thank you so much for clarifying that. I think it's great that you've done such a thorough job of working in such a complex and dynamic sort of data set. So thank you for being able to speak to that a little bit more. That was all from me.
If there are no more questions from the judges, we'll open up questions to the audience. And so if you do have a question, please type it into the chat or raise your hand so that I can unmute you. We do actually have one question already in the chat. And the question says, "Hi Gemma, fantastic presentation, very insightful and easy to understand. I learned a lot. I'm wondering about the applicability of this research. Is it enough on its own to be able to predict the climate next year? Are we able to project it a certain number of years into the future? And could we modify your approach to account for climate change and ocean warming, etc.?"
That's a big question.
Yes,
Okay.
you can feel free to answer.
Thing 1, even if you could predict ENSO perfectly, the best estimates that give you for understanding the rainfall is understanding about 20% of the rainfall. Rainfall's really complicated. Rainfall's really random. Weather patterns influence so much of what any rainfall is. So all understanding ENSO does is it gives you an idea of what sort of rainfall range you're looking at for the next year. Part 2, if you want to predict ENSO out into the future, the best you can do with a really complicated big model that you run on the supercomputer is about 9 months, maybe 12 months if you're really lucky and conditions happen to be favorable for the model actually behaving like it does and you get less randomness coming in. And you definitely can't do that with this simpler model. But what the simpler model does is it gives you more of an understanding of what the behavior of the system is in order to perhaps understand better and improve your more complicated models. Was there a third part?
The third part said, could we modify your approach to account for climate change and ocean warming, etc.?
Sort of. So I guess part of why I was looking at how the parameters change over time is to see if there is this shift with climate change. And there are other like mediumly complicated models where you can adjust climate change a bit and see what happens. And then there's the really complicated models, which just break down all the little tiny physical components. You can say, look, it's now 2 degrees warmer, run the model, see what happens. And they all disagree about what happens to ENSO. So there are ways of looking what will happen in the future, but it's not entirely clear yet how it works. So you need to look at the complicated models and the simpler models as well. Does that answer the question?
I think so. There are a couple more questions, but we've run out of our time for Q&A. And I think you've answered most of them in your previous discussion. I'm just going to let Sylvester has joined us. I'm just going to try and see if we can. There we go. So Sylvester, are you ready to present?
Hello. Yes, I am. I'm so sorry for this mix-up. There was something wrong with my laptop update. I just updated yesterday. wasn't working too well, but I'm ready now.
No worries. Do you have your, awesome, wonderful.
Hello.
Yeah, so whenever you're ready to begin presenting, you'll have 10 minutes followed by 5 minutes of Q&A.
Awesome. Okay, let me just put my slides up then. Hello everyone, good afternoon. I'm Sylvester. I'm a final year undergraduate from the National University of Singapore, and I study biology here, and I'm going to talk about urinary tract infections today. So the term urinary tract infections might sound very familiar to most of you, and some of you might have even experienced it before. In fact, it's estimated that 50% of all women around the world will experience UTI at least once in their lifetime. And more importantly, UTI actually has very wide implications for healthcare systems all around the world. Annually, UTI incurs up to $1.6 billion in expenditure in the US, and that's 1% of Australia's annual healthcare expenditure. And we're talking only about a single disease here. With aging populations all around the world, we are expecting to see more cases of UTI, especially amongst women, because women above the age of 65 has 2 times higher risk of getting UTI than women under the age of 65. And even amongst young women, UTI can be very serious because it can cause preterm labor, which can cause very, very serious complications in pregnancy. Now, surely a disease like UTI must be caused by something, right? There are many organisms that can cause UTI, but I've just decided to just mention a few of the top candidates. So there's Klebsiella pneumoniae, Escherichia coli, as well as Staphylococcus saprophyticus. Now, they do have some really enigmatic names here, but the focus here is actually on E. coli or Escherichia coli here, because it is responsible for up to 75% of all UTI around the world. So now that we know the severity of the disease, and we also know the culprits of UTI, how can we defeat it? And one answer to this question lies in genomics. So what is genomics? Genomics is more than just a buzzword. It's the study of all the DNA that exists in all living organisms as well as viruses. It doesn't matter whether it's a fish, an insect, a plant, or a virus. All of us have genomes, and the genome provides us with the blueprints for various functions that encode life. And this includes in E. coli, which causes UTI. E. coli also has a genome, and this genome provides the instructions for E. coli to cause UTI. So how can we exploit genomics to understand and treat UTI in this case? If genomics is the complete set of DNA found in a living organism that encodes life, then the answer to UTIs caused by E. coli should also be found in its genome. Now, I'd like you to imagine the genome as a book, and just like any book, there are pages in a book. Inside the pages of this genome, every page can have a different instruction. We can have pages that teach E. coli how to move, pages that teach E. coli how to reproduce, but we could also have pages that teach E. coli how to cause UTI. But what I just showed you were just 3 pages. In reality, actually, we have over 5,000 pages inside E. coli's genome. And in these 5,000 pages, any of these pages, or a combination of these pages, can encode the factors that allow E. coli to cause UTI. And this is precisely where genomics gets extremely powerful for understanding UTI. I'd like you to imagine this scenario where we have a non-pathogenic E. coli, and I will call it the cousin E. coli here. And in this cousin E. coli that doesn't cause UTI, it also has a book of instructions. And since the genome encodes all instructions, in theory, we could transfer random pages from the E. coli that causes UTI into its non-pathogenic cousin. And we should see that some of these pages will give the instructions for allowing this non-pathogenic E. coli to cause UTI. And that's precisely what my team has done. Now imagine that these transfers are done, and we did these random transfers multiple times of different cousins of the pathogenic E. coli. Here we have 3 cousins, and all of them receive different pages and a different combination of pages. Remember that these bacteria originally do not cause UTI, but because of these pages that they receive, there's a chance that they could now cause UTI. So we could run some tests and we can see that, oh, most of these cousins do not actually cause UTI, but that's okay. Because the moment we find one that causes UTI, we can look into his genome and look for the exact pages that were transferred. And we know that this page is likely the cause of UTI. And that's what my team and I did. So here's a graph which looks very complicated at first glance, but let me just walk you through it. All the red bars that you see here refers to a cousin that does not cause UTI originally. And the focus are on these asterisk signs here, which shows that these clones, particularly these 3 red bars here, have a statistically significant ability to cause biofilms, which is very, very highly strongly indicative that it can cause UTI. If you were to go back to the book and pages analogy that I just went through before, I'll just run through this figure again. On the left axis here, you see that each row here corresponds to 1 different pathogenic cousin. All of these rows are different. For the bottom axis, it refers to the entire book of the non-pathogenic E. coli. You can imagine that this is a book, and from left to right, it goes from the first to the last page. And each of these red and blue bars that you see here corresponds to different pages that were transferred. So from this diagram here, you can see that on the y-axis, as you go up, every single bar differs, and therefore every single cousin here has a different page. And if you look at these 3 here, which I colored in red, these are the ones that are similar to the one in the previous slide with the asterisk signs. In other words, these are the ones that are able to cause a significant amount of biofilms. And we believe that these clones here, these cousins which receive these pages, are likely able to cause UTI. So to conclude this part of my presentation, we know that E. coli causes over 75% of all UTI cases, and there are over 5,000 pages in its genome, of which any combination of it can cause UTI. By randomly transferring these pages to a non-pathogenic cousin, you can find that some of these cousins actually receive pages that makes it acquire aggressive behaviors often seen in UTI-causing E. coli. When we look into the genome of these transformed cousins, we can identify the specific pages which were transferred, and we know that these pages inside this genome are likely the instructions for causing UTI. But wait, the quest is still not over yet. If we look back at this diagram here, you'll notice that there are some gaps in the pages that are transferred. In fact, all these bars here account for up to 88% of all the pages that exist, and there are still 12% which cannot seem to be transferred. Now the big question here is, what if the factor for causing UTI is found in this 12% here? Then, at present, we are unable to check for these pages. So what I did was to try and increase the amount of pages that are transferred. At present, we were at 88%, but I increased it to 95% by using specific donors and specific recipients. In this case, I selected specific UTI-causing E. coli and transferred it to specific non-pathogenic E. coli to receive these pages. And we saw that there was a 7% improvement in the pages transferred. However, there's still 5% left, and we are still trying to find out why this 5% cannot be transferred. But one thing we believe is that these 5%, when they are transferred, will cause the non-pathogenic cousin to not be able to survive. So to conclude, today I've spoken about UTI and its healthcare burden around the world. We know that it causes a lot of problems to women who experience it, and it also causes a significant amount of healthcare expenditure. We have seen that genomics has the potential to answer crucial questions about UTI, and we are living at a time where genomics is at its cutting edge. And these findings should theoretically allow us to come up with new treatment methods and prophylactic methods to prevent and treat UTI. So to all of you who are listening today and might have experienced UTI, I hope you remember my talk today and remember that there's a team of scientists like myself, and we are working very hardly to try to understand how UTI is caused and find ways to treat it. Thank you.
Thank you very much, Sylvester, for that awesome presentation. There are now 5 minutes for Q&A. We'll begin with our judges. Do you both have any questions?
I can start. Thank you so much, Sylvester. I just wanted to take the opportunity to ask, I might have missed it because you only touched on it briefly, but could you just expand a little bit more on that second process that you had to pick up that missing 12% in that genomic information? How was that? Was that significantly different to like that first process you had, I guess, to like collate those pages and try and see where that information was? Like, was that a little bit different to what you did initially, or? Yes, I'm not sure if you spoke to that.
Right. So the reason why we're observing this 88%, there are these 12%, is that the reason why it's only at 88% is because there are many instances in which the same pages are being transferred. And the reason for that is because the way we set up this technology, there are inherent biases in the technology that we are building on. And this technology is known as transposon mutagenesis. In essence, when we try to transfer pages, we need to put something inside the genome which allows this part of the genome to be mobilized. And when we put this thing, you can think of it as like a ticket. These tickets tend to be concentrated in specific parts of the genome. And so when you have an overrepresentation of the tickets in certain regions of the genome, it will just keep retransferring the same parts. And so just by numbers, we are prone to observe repeats of the same thing. So to overcome this problem, I specifically chose individual pathogenic E. coli where the tickets are placed in the areas which are underrepresented. And when we have the tickets in the underrepresented area, it allows us to force the system to transfer regions that have not been transferred yet. I hope that answers your question.
Yeah, thank you so much. That was a great way to clarify it. And I'd particularly like to commend the way that you've taken such like a complex process and tried to make that process really clear and easy for us to follow. So thank you.
Thank you.
Okay, if there are no more further questions from the judges, we'll open up the floor to the remainder of the audience. If you do have a question, feel free to just type it in the chat or raise your hand so that I can unmute you. So yeah, Veronica, feel free.
Yeah, if that's okay. Sylvester, I just had a question regarding one of your graphs. I think it was one of the earlier slides, or the first graph that you showed us.
Right.
This one.
Mm-hmm.
I was just wondering, so with the clones where you have the asterisks on it, mainly the two highest ones, could you, I guess, just address the bars, the error bars?
Right. So the error bars here are standard deviations, and unlike standard errors or means, they are definitely a lot larger than you would expect. And the reason why we see such high variabilities, as you observe that the variability here is a lot less, is we believe that because of the media that was used to grow this bacteria and force it to express this thing that we call the biofilm, the phenotype that we are observing is highly variable inherently because there are extremely many genetic factors that affect biofilm formation. And what I mean is, when we look at the media that is used, there are many minor things that can change. It's almost like a butterfly effect. So if I were to use a media today that is 2 days old, and tomorrow I repeated the same thing, exactly the same, but my media is 5 days old, you could see like a huge fluctuation in the biofilm formation. And so we had to do replicates like that to account for these fluctuations and to show that even with these fluctuations, the distributions are significantly different such that we know that there's an uptick in the amount of biofilm formation. So in essence, the reason why it's highly variable is because there are extremely many factors that affect this phenotype that we are observing. And it's impossible to fully control all of them as of now until we have further clarity on this phenotype. I hope that answers your question.
Yes, it does. Thank you.
Awesome. Well, that brings us to the end of the 5 minutes of Q&A. Congratulations on the presentation again, Sylvester.
Thank you.
And that also brings us to the end of today's session. So I'd like to thank our judges, Christy Davis and Eileen Walsh, and also a huge congratulations to all of our student presenters. Each of your presentations was really insightful and really interesting. So just wanted to let you know also that this recording will be made available online following the conclusion of this conference. And I hope to see you in the other conference sessions that remain throughout this afternoon. So thank you very much, everyone, and a job well done.
Thank you so much, presenters. You did a great job. Thank you so much.